Chapter number 2, Biological Classification. 2.1 Monera, Kingdom Monera. 2.2 Kingdom Protista. 2.3 Kingdom Fungi. 2.4 Plantae. 2.5 Animalia. 2.6 Virus, Viroids and Lichens. In this part we will discuss Kingdom Monera. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, two-kingdom system of classification with Plantae and Animalium kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphological, a need was also felt for including other characteristics like cell structure, evolutionary relationship, etc. Classification system for the living though plant animal kingdom have been a constant under all different systems. The understanding of what groups organisms be included under these kingdoms have been changing. The number and the nature of other kingdoms have also been differently by different scientists over the time. R. H. Whitaker, 1969, proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of the five kingdoms. Table 2.1 is discussed by me in a short. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain. Thereby, a six-kingdom classification. You will learn about the system in detail in higher classes. Let us look at this five-kingdom classification to understand the issues and considerations that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cell. This placed together groups which wildly different in other characteristics. It brought together the prokaryotic bacteria and blue-green algae cyanobacteria with other group which are eukaryotic. It also grouped together the unicellular organisms and multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and autotrophic green plant, though they have showed a characteristic difference in their cell composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characters were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas chlorella, earlier placed in algae within plants, and both having cell walls, and with Paramecium and amoeba, which were earlier placed in the animal kingdom, which lack cell wall. It has a put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characters and evolutionary relationship over time. An attempt has been made to evolve a classification system which reflect not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, i.e., is based on evolutionary relationships. In the chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whitaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdom, respectively, will be tapped separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrio, and spiral spirillum. Though the bacterial structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolical diversity. Some of the bacteria are autotrophic. For example, they synthesize their own food from inorganic substrates. They may be phototrophic, autotroph, or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. They depend on other organisms or dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty area, halophiles, hot springs, thermoacidophiles, and marshy areas. Methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow and buffaloes, and they are responsible for the production of methane biogas from their dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A, similar to green plants, and are photosynthetic autotroph. The cyanobacteria are unicellular, colonial, or filamentous, freshwater, marine, or terrestrial algae. The colonies are generally surrounded by gelatinous sheet. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyst, nostoc, and anapenna. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Chlorella, typhoid, tetanus, ceteris canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions, they produce a spore. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the another. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. For diagrams, you can check out our short. Thank you. Like and subscribe for next part of Protesta. Bye bye.